퀴즈는 준비되어 있지 않고요. 네, 큰 박수 부탁드립니다.
Hello everyone. Today we're going to present about the analysis of DNA storage-based DBMS and suggestion for its development. As you all know, as the modern society, which is also known as the sea of the new information, progresses, a huge amount of information is being generated even at this moment. Then, what will happen in the future? What will happen in the society from now on? According to the Data Age 2025, the world's total data scale will grow 10 times more than it is today to 163 zettabytes. This is why people have been thinking that we will need a new form of data storage in order to handle this amount of data. And to that end, people have been evolving their own type of DNA data storage. And today's topic, the DNA storage, is a concept that had emerged from this background. Our study's goal is two things. Firstly, to find out which database management system should be in place before the commercialization of DNA storage. And secondly, to suggest the features of it before which fits the DNA storage. So let's take a moment to see what exactly a DNA storage is. A DNA storage is, in short, it is a line of data that is stored in the DNA. If I told you just like this, you might wonder how does it really works? Although the encoding system varies from lab to lab, the basic framework is as follows. The DNA synthesizer, and the PCR thermocycler, and the DNA library, and the DNA sequencer. To explain it more specifically, these processes are done by as follows. First, the encoding system. In the encoding system, binary codes such as 0001, 10, and 11 are represented as the nucleotides such as A, G, C, and T. And next, during the synthesized storage retrieval and the sequence process, we cut the DNA storage piece into the pieces, and then we save it, and then we make a back of it with a contribution. And the final step is the decoding process. In the decoding process, we turned the nucleotides back into the binary codes such as 0001, 10, and 11.
Then let's look at the step of DNA storage that lasts over 10 years. The first of research is 2007, the research that made the device which is using addressing molecules to mismatch sites with DNA strand. The next one is 2012, with the science published the research that encoded DNA with digital information that includes HTML draw a word book, 11 image files, and also 1 JavaScript. This research showed us that DNA storage can be another type of DNA storage medium, just like hard disk or magnetic tapes. The third one is 2013. The innovation of 2013 is using error connecting encoding schema, which is ensured to extremely lower loss data rate. The last one is 2018, which is the newest one. The research proposed and evaluated the new technology of random accessing of DNA storage. To draw conclusion, we should analyze the pros and cons of DNA storage, and we should find out what is suitable for DNA storage, just like DBMS. So we found that advantages and disadvantages of DNA storage, or pros and cons of DNA storage, in this screen. We found that DNA storage is unstructured data and also big data. RDBMS, which is first appeared the DBMS type, normalized data to table of columns and rows. So it has exit attributes and gives data integrity. However, unstructured data is increasing, so fixed format of DNA strand is invalid. So NoSQL is appeared that is more flexible. So NoSQL has easily scalable and easily controlled to big data. We suggest that NoSQL, or just called NoSQL, is suitable for DNA storage, not RDBMS. Our research that we conducted is pretty significant because we made the background of commercialization of DNA storage, and also we give a path for junior who are studying similar topics just like us. However, our research is just only based on document, so we didn't proceed research of our recommendation. So we are going to try to find out more detailed conclusion and try to conduct our research more deeply. Thank you for listening.